Form 4 Biology Transport in Plants Part 2. You can find this lesson in Chapter 11 in your student book and the pages 159 to 163. Objectives of this lesson: to know about transpiration, to understand the factors affect on transpiration, and to know the ways of measuring transpiration. Let's learn about transpiration now. Transpiration is the loss of water vapor from the leaves. As we know that water loss from the stomata. Water leaves the cells of the leaf mesophyll and evaporate into the air spaces. Then this water vapor diffuses out through the stomatal pore. We can see this one as transpiration. Transpiration causes transpiration stream. We know that transpiration is the loss of water from leaves. This results water potential gradient. So xylem vessel always try to fill that gap, providing water to the leaf mesophyll cells. So transpiration causes water to be pulled up the xylem in the stem and roots in a continuous flow, known as the transpiration stream. Let's read once again. Transpiration causes water to be pulled up the xylem in the stem and roots in a continuous flow, known as the transpiration stream. What are the functions of transpiration stream? First one, supplies water for the leaf cells. This is very important as leaves undergo photosynthesis. We know that water is a raw material for photosynthesis. Second one, carries mineral ions dissolved in water. Roots absorb mineral ions from the soil. From the roots to the leaf and to the other parts, they need to transport. This transportation done by transpiration stream. Third one, provides water to keep the plant cells turgid. We know that turgid state, when the water enters to the plant cells, they become turgid due to internal pressure. This is very important to plants to keep upright. Fourth one, allows evaporation from the leaf surface. This is important to cool the leaf. It is very similar to the process of sweating in human. Of transpiration. There should be a control of transpiration, otherwise plants will suffer from water loss. More stomata on the lower surface of the leaves than the upper surface. This is one of the adaptations. There are two surfaces, upper surface and lower surface. In that lower surface, they contain more stomata than upper surface because upper surface is always facing to the sun. Then there's a control. Another one, stomata can open and close. This opening and close done by guard cells. You can see that diagram, stomatal opening and guard cells. These guard cells are control the size of the stomatal opening. So whenever they need, they can control by opening and closing. What happened to the stomatal pore in the light? In the light, water enters to guard cells by osmosis because they need to undergo photosynthesis. So water is a raw material. Then guard cells become turgid. When they become turgid, they swell up and bend outwards. You can see the picture. They bend outwards due to the turgid state. So what happened to the stomatal pore? Yes, stomatal pore opens. So in the light, stomatal pore opens. Tell me what happened to the stomatal pore during night time or in the dark. Can you suggest? Okay, let's see. Guard cell lose water and become flaccid. So when they become flaccid, what happens to the stomatal pore? Yes, stomata closes. You can see in the diagram, they become flaccid due to loss of water. So stomatal pore closes. This is very important because without the sun, there's no need for loss of water from the stomata to cool the leaves. As a one use of transpiration stream, we learned that transpiration is very important to cool the leaf because when the water evaporates, it uses the heat from the plant. But in the dark, they don't want to cool the leaves, so there's no need of loss of water. In addition, leaves cannot photosynthesize in the dark because there's no sunlight, so they don't need water. So no need water to enter to the leaf in the night. Rate of transpiration changes time to time due to certain factors. What are those factors? First one, light intensity. Can you remember we learned that in the light, stomata open. Why is that? Because we know that they receive water to undergo photosynthesis. So what happened to that cells? Which cells? What are the cells? Around that stomata? Guard cells. So they become turgid and bend outwards. So because of that, what happened when the stomata open during the high light intensities? What happened? Yes, rate of transpiration increases. So the rate of transpiration increases in the light. Second factor, temperature. What happened during high temperatures? We know that in high temperatures, the rate of evaporation also increasing. So this results increase the rate of transpiration. Third factor, humidity. What is humidity? The amount of moisture in the atmosphere. So you can see that humid condition. Now in the humid condition, when the air around the plant is humid, this reduces diffusion gradient between the air spaces in the leaf and external air. Now we know that around the leaf also there are water moisture or water vapor around there. So in the leaf also there's some water content. So the diffusion gradient is less. Because of that, the rate of transpiration decreases in humid conditions. Fourth one, wind speed. Now what can you tell about the transpiration rate in windy conditions? What happened to the moisture content or the water vapor around the plant during the windy condition? We know that transpiration increases because of the fast air movement removes water vapor near the stomata. With the wind, water vapor around the plant moves away. So this results high diffusion gradient, and it finally causes high rate of transpiration. Now we know about the transpiration. The rate of transpiration can be measured using a potometer. What is a potometer? It's a simple piece of apparatus which measures the rate of transpiration. There are two types of potometers. First one, weight potometer. Second one, volume potometer. Let's learn about those potometers in detail now. Weight potometer. You can see a weight potometer in the image. This measures the rate of loss of mass from a potted plant. You can see that there's a polythene bag to prevent loss of moisture by evaporation from the soil. Why do we need that one? We know that when we are keeping that plant to check for the transpiration, there will be loss of water from the soil too due to evaporation. But if we take this as the final measurement, it gives false reading because we need to measure the loss of water only from leaves. Okay, so we are covering that soil using a polythene bag. We need to get the initial mass of a potted plant. Then we have to leave for certain time period, allowing it to transpirate, and finally we need to get the mass. By using this equation, you can find the rate of transpiration. Final mass minus initial mass divided by time period. Okay. You can see that control. When we are measuring the rate of transpiration by using that weight potometer, we can use a control. Control means without the tested factor. Tested factor is the plant for transpiration. So we can set up a control without a plant. So by using this control, we can exactly tell that the loss of mass is due to loss of water from plant leaves. Volume potometer. You can see a volume potometer in the given picture. We can use to find the rate of uptake of water by a leafy shoot. In this volume potometer, we assume that rate of uptake of water is equal rate of loss of water. So we can find rate of uptake of water. You can see that leafy shoot is there and it is connected to the beaker of water through a capillary tube, and there's a air bubble. Okay, we are allowing that leafy shoot to undergo transpiration over some time period. Then we measure the distance moved by the air bubble. You can find that of transpiration by using this equation, distance moved by the air bubble divided by time period. This is a simple volume potometer. You can see that leafy shoot is connected to the rubber tubing and to the capillary tube, and these all apparatus submerge in the beaker of water, and there's a air bubble trapped in the capillary tube. When we are setting this apparatus, we need to follow these. The capillary and rubber tubing is placed in a sink of water. air is removed by squeezing the tubing. The end of shoot is cut at an angle. This is to make it easy to push into rubber tubing. And we need to seal the joints properly. The distance moved by the column of water in the capillary tube is measured over a certain time. We assume that uptake of water is equal to the loss of water. So we have to measure the uptake of water by using the movement of water column. So we can measure the rate of transpiration by distance moved by the air bubble divided by time. The plant can be exposed to different environmental conditions to see the effect. Now we learned that there are certain factors that affect on rate of transpiration. If you want to check that rate of transpiration in different conditions, we can use these apparatus. For example, we can see what happened to the rate of transpiration during the windy condition. To do that one, we can expose this apparatus to the fan. When we are keeping a fan, it is similar to the windy condition, so you can check what happened to the rate of transpiration. Or else you can keep under a light and see what happened to the rate of transpiration. From that one you can guess what happened to the rate of transpiration during high light intensities. Likewise, you can check rate of transpiration in different conditions by using that apparatus. Now we are at the end of the chapter 11. We already completed this lesson in Transport in Plants part 1 and this Transport in Plants part 2. Okay, now you can complete questions 4, 6, 7, 9, 10 in pages 164 to 167. You can watch some YouTube videos to get the further knowledge. Hope you understood the lesson very well and stay safe. Thank you.